Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic that is your quality control in the dairy industry. Okay, we all know that this dairy industry are what? They are they produce they have a milk and milk product. Okay, in dairy industry what we have manufacture or what we have process, we process a milk and a milk product. Okay, now this milk and milk product both are what they are come under a perishable products. Perishable products means that their shelf life are very less and moisture content is high. When the moisture content is high, then the bacterial load will increases. Bacterial load will increases. Okay, and that's why their shelf life is very less, because when a moisture content is high, then it is a higher chance that the bacteria can easily grow into this type of product. Either it is a milk or a milk product, and it will uh, spoil them. Okay. So why it is important that we have to discuss the quality control in a dairy industry? Because of this perishable nature. Okay, now what is the meaning of this quality control? We all know that this quality control is what? Maintaining the quality of the product, that it is at their standards or not. Okay, so at last, suppose that we are going to manufacture a paneer. Now we check this quality, like what is the moisture content of that quality, sorry, that paneer, what is the TLC count, like total plate count, TTPC, what is the bacterial load, everything. Okay, what is the ash content, what is the fat content. So when we check the quality of the end product, then it will come under a quality control, that our product which we get after the manufacturing is fulfill the demand or not, or it is up to the standard or not. Okay, so basically when we do this quality control, then it can measure in a farm practices. From practices can affect the quality control in a dairy product: milk collection, processing, testing, packaging, labeling, hygiene. Everything will affect the quality of the dairy product. Okay, if we talk about the farmhouse, then we all know that that farmer who are milking the milk should be milk in a proper condition, like their hands should be clean, and they have to follow the government guidelines for the milking purpose. They have to follow the government guidelines. Okay, now milk collection is the another way. We all know that suppose that the milkman who are collecting the milk, he was suffering from some disease. So maybe the contaminate like contamination disease. What happened? Sorry, communicable disease. So these disease will pass through into a milk, and along with the milk, it will also transfer to the another person who consumed that milk. So milk collection should also be done in a proper hygienic condition, and it will also affect the quality of the dairy product. Now after milk collection, that is your processing. Suppose that in a processing, we all know that we have to what, like for a milk processing, suppose that for a milk processing we have to do a pasteurization. Okay, Now the proper temperature will not follow. Then in that case also it will affect the quality of the dairy product. Now next is your testing. We all know that when we have a raw material and we have a product, okay, these both should be like tested for both, for a microbial testing and for a chemical testing. Okay, packaging means that suppose that the proper packaging material are not used, or maybe the packaging material that we are use is like already contaminated, so that's why it will also affect the quality of that dairy product. Labeling. The next is your labeling. Suppose that we what we have to do, we have to label a proper labeling, like we have to mention a nutritional value, we have to mention like your allergens. Okay, so whole information should be given in that product. And the last one is your hygiene. Hygiene should be maintained when we are working in a any food industry, but especially in a dairy industry it is very important. So that's why we are going to discuss about all these things, that is a quality control in a dairy industry, or how we control the quality of the dairy product. Okay, so we only take the end product, because here we are talking about the quality control, not the quality assurance. Okay, there is a difference between the quality control and quality assurance. Okay, quality assurance means that we maintain the quality in each step. Maintain the quality in each step. But in quality control, what we have to do when we get a product, then we check it that the product is in our standard or not. Okay, so let's start today's lecture. Introduction. The quality of the food, such as milk and dairy product, may be defined as the sum of the characteristic which enable the food to satisfy definite requirement and which determine its fitness for a consumption. Okay, when a word arises, that is your quality of the food, then what does this means? What does this means? Okay, suppose that if we talk about the quality of the milk and milk product, so simply it is defined as the sum of the all the characteristic that should be present in that particular product which satisfy or which fulfill the requirement of the like requirement, that is like that is your standard, which fulfill the standard, and it also fit for the consumption. That is called a quality of the product, that satisfy the standard, and along with the standards it also fit for the consumption. Okay, and this definition is given by a Molnar in 1993. Molnar in 1993. Okay, now the next is your, in this sense, quality can be judged by means of sensory evolution, its nutritive value, and according to its chemical, physical, and microbiological characteristic. Okay, now if we have to like judge, or if we define the quality of the product, now it can be divided in the sense of either, like no, not this either, it is the combination of sensory evolution, nutritive value, chemical, physical, and biological characteristic. First of all, what we have to do in a laboratory, we perform all these tests, like nutritive value. We check it that suppose that any product like in the milk or milk product, so what we have to do, we Check the nutritive value like its carbohydrate concentration, its fat, protein, and along with it, vitamins and minerals. Okay. Then the different microbiological characteristic like your uh, total plate count, okay, clostridium, E. coli, okay, chemical characteristic like your presence of like any chemicals or anything, okay. Now sensory evolution means that suppose that we prepare a cheese. Now we have to take a sensory evolution of that cheese, that it is fulfill the like standard of the sensory of the cheese or not. And we also check the physical characteristic of that product. When referring when referring more especially to the microbiological characteristic of a food, it implies the measurement of the hygiene quality of that product. Okay, so along with the microbiological characteristic of the food, we also measure the hygienic quality of the that particular food product. The concept of hygienic quality in turn required that undesirable microorganisms or residue do not gain access to milk or dairy product. Okay, so when we, what is the concept of this hygiene quality, why we maintain the hygienic quality? Okay, so the main purpose of the maintenance of hygiene quality of the dairy or milk or dairy product, that it can require sorry, it can not not gain the undesirable microorganisms or residue in. Okay, suppose that we all know that it is said that uh, E. coli should not present into any product. Here I am talking about the milk. Now, how it is not present, that we have to fulfill or follow the hygienic quality. Okay, we have to avoid the cross contamination. We have to maintain the like utensil which are used in a like your processing industry. Your personal hygiene should be maintained. In that case, you will maintain the hygiene quality, and it will affect that the undesirable microorganisms or residue do not present in a excess into a milk or a dairy product. Okay, if it is present, then either it will affect the human health or cause a spoilage or deterioration of the food, or sometime maybe it also become a reason of the death of the human being. Okay, that's why we maintain the hygiene quality, because these may prove harmful to human health. This e coli is very danger for the human health. That's why no single colony should be present into any food. No single colony should be present in any product. Cause spoilage or deterioration of the food product, or simply be frowned upon from an aesthetic point of view. Okay, now this is about the introduction of this slide. Now come to the next one. Cases of different foodborne diseases and food poisoning are become more and more common. Okay, cases related to the foodborne disease and food poisoning. Okay, now these two things are very different. Okay, foodborne disease means that the disease which arises due to the eating or consumption of contaminated food and water. Okay. Now, this food poisoning means that we take a food which are already spoiled by the toxic of the bacteria. Okay, it is arises due to the consumption of toxic produced by a spoilage microorganism into the food product. Now, we all know that these cases are becoming more and more common throughout the world, and it will ultimately affect the public health and also raise to the microbiological spoilage. Okay, why these things are happening? Because of the ignorance and carelessness. Okay, these two things are the basic reason for this foodborne disease and food spoilage. We all know that the food should not kept at a danger zone for 2 hours. Okay. Now, what is this danger zone? Means that the temperature range between 5 to 63 degrees Celsius. So either we have to keep the food above 63 degrees Celsius, that is your hot temperature, or below 5 degrees Celsius, that is your cold temperature. Then we then we protect the food from the microorganisms. Okay. Now, if it is not follow, then obviously the microbial attack are occur and it will spoil the food product. And when the food product are spoiled, then obviously when it is consumed by a consumer, then again it will affect them. Now, this microbiological spoilage occur in any stage. Now, what does this mean? Now it is occur into a raw materials during a cutting, no, not a cutting, during a preparation. Now, in preparation, there is a cutting, grinding, and all these things are cut occur during a processing. Okay. Now, during the storage. Now, during the packaging. It will occur in any time. Okay. When these microbiological spoilage are occur, ultimately it will affect the product, affect the end product. Now, this end product are consumed by the consumer. Now, if they consume it, they will affect the health of the consumer. So that's why the cases of this and a a food poisoning are increases. This is the reason. Now, see what is written in a slide. Cases of foodborne diseases and food poisoning are becoming more and more common throughout the world. Both of these public health problem and the microbiological spoilage of the food can be minimized by carefully choice of raw material and correct manufacturing and storage procedure. Now, how we minimize all these things? When we have correct these things. Okay. Then ultimately the microbial spoilage will also correct. Achievement of such objective required in many cases monitoring at various stages to access a microbiological load or to look for a particular microbial type. So for achieving this object, what we have required, either we have to monitor the microbiological load in every stages, and along with it we also know the microbial types. Okay, so in the next slide we are going to discuss about all these things. Now this in this slide we are going to discuss about the control of airborne microorganism in dairy industry. Okay, now here what should we are going to do? We are going to do that. What is this airborne microorganisms? Okay, means that the microorganism which are present into the air. Microorganisms present into the air. So here we are going to discuss about that, how we control these type of microorganisms. Now the demand for extended shelf life and safety of dairy product has put increased emphasis on the microbial quality of air in dairy environment. Okay, so if this shelf life extended of shelf life and safety of dairy product, it's totally depend upon the microbial quality of air in a dairy environment. Okay, if the dairy environment contain a higher microbial load in their air, then obviously it will affect the quality of the dairy product, and ultimately it will affect the shelf life of that product. So we have, first of all, what we have to do, we have to maintain the microbial quality of the air in a dairy environment. Air quality in the processing and packaging area is a critical control point in the processing of dairy product. Okay, so air quality in the processing area and the packaging area is the critical point in the processing of dairy product. Okay, when we are processing the dairy product, so the two area, that is your processing area and the packaging area of their air, are very critical control point. Okay, for maintaining the quality of the air at that area, that is your processing and packaging, it is a critical control point. Why? Because air contamination reduces the shelf life and may serve as a vehicle for transmitting the spoilage organisms. Okay, again, I already told you this airborne contamination, what they do, if the air is contaminated, then ultimately it will reduces the shelf life, and also it will transfer the spoilage microorganisms. Okay, and suppose that if there is a pathogenic microorganisms are present in that air, so this pathogenic microorganisms will transfer the disease to the person. Okay, now this is said by a anon. This will taken by from a anonymous 1988 Kang and Frack 1989A. For every precaution should therefore be taken to prevent a airborne contamination of the product during and after the processing. So every type of precaution we have to take so that we prevent the airborne contamination, and along with it also we have to uh, sorry we have to protect the airborne contamination of the product, either before the processing or during the processing or after the processing. Air quality in processing area, the factory environment, and air used in the manufacturing of the dairy products should be monitored on a regular basis. Okay, so air quality in processing area, factory environment, and even the air which are used in a manufacturing of dairy product, all these type of air quality should be monitored in on a regular basis. Airborne microorganisms in a dairy plant like your bacteria, mold, yeast, and viruses. These are the microorganisms which are present into our air, uh, which are present in a dairy plant. Now, the next is your data on air count and types of different microorganisms in processing area reported by the author are comprehensively reviewed by a Kang and Frank in 1989-11. Okay, so there are the different data and the air count and the type of the microorganism which are present in a processing industries. All these are reported in a various author, and they are comprehensively reviewed by a Kang and Frank in 1989. The generic composition and the level of the microorganism can vary widely within and among the plants and on a day-to-day basis within a same plant. Okay, even the generic of the composition of different microorganisms are also vary even within a plant and among the plant and day-to-day basis on the same plant. The variation can consequently be attributed to the differences in plant design, airflow, personal activities, and status of factory hygiene. Installation of air filter, application of the UV radiation, and regular chemical disinfection like uses of the bactericidal, fungicidal, and virucidal agent of air can be applied to a critical area so that it will control the airborne microorganisms. Okay, if we install the air filter, then again it will control the airborne microorganisms, UV radiation, regular chemical disinfection like bactericidal. Bactericidal is used for the elimination of the bacteria, fungicidal is for the fungi, and virucidal agents for the viruses. So all these things that when we uses, then mm, it will control the airborne microorganisms. Now the next is your sources and roots of airborne microorganisms. Airborne microorganisms can be attached to a solid particles. Okay, these airborne microorganisms, they are attached to a solid particles like your dust. Suppose that this is your dust. Now this dust are contain a tiny airborne microorganism which cannot be seen by our naked eye. Okay, are present in aerosol, droplets, or also occur as a individual organisms due to the evaporation of water droplets or growth of certain mold species. The main source of this airborne microorganisms are the activity of the factory personnel. Okay, the person who are working inside the factory, ventilation and air conditioning system. They are what? They are the main source of the airborne micro or microbes in follow of outdoor air and packaging material. It is said by a Heldman et al. 1965, Hedrick and Heldman 1969, Hedrick 1975, Kang and Frank 1989b, Luck and Gravon 1990, Frontini 2000s found that factory personnel, daily equipment, building material, and ventilation system are responsible for the 50 to 60%, 25 to 35%, 10 to 20 YO, and 1 to 5 YO respectively of the airborne contaminants. Okay. Now the next one is your an increase in viable aerosol have been detected during the flooding of floor drain. Okay, if there is a increase in viable aerosol, then it can be detected by a flooding of the floor drain by a Heldman and Hedrick 1971. And after rinsing the floor with the pressure of water hose, thereby illustrating the ability of the microorganism that can be disseminated from the drain and wet surfaces, which is a result of the physical activities. By Kang and Frank 1990, Mettler and Carpentry 1998. Whenever possible, wet cleaning should not be used during the processing of the milk product in areas in which the product is exposed and can be contaminated by the aerosols. Okay. Once there is a high concentration of this viable aerosol, it will generate it. It can take more than 40 minutes to return to the normal background level. It is important to minimize the generation of the aerosol droplets from the bubbles bursting at a water surface, for example, during rising activities or during a raw milk handling. It also shows that the drain, floor, and standing or condensed water also a source of the pathogens in a dairy plant. Eel Shenave 1998. The potential contamination from the bacterial biofilm is a major concern. Okay, so potential contamination which is created by a bacterial biofilms are the major concern because microbial cells attach, grow and colonize on open exposed wet surfaces. Okay, when there is a wet surfaces, then this microbial cells attached into it. So to suppose that this is your wet surfaces. Now here it is a microorganism. Now microorganisms are attached here. Now after attaching, it will grow and form a colonies when it is exposed to a wet surfaces. For example, like your floor drain, walls, conveyor belt. Okay. Now the next is your water which are used in open circulation system is another significant sources of airborne microbial populations. Okay. Again, water is also important source for the airborne microorganism. Okay, it is taken by a Lightard and Frisch 1976. These authors found that as many as around 10 viable bacteria per second can be released into the air from a 50-meter high cooling tower. Okay, from a 15-meter high cooling tower, there is around 10-fold viable bacteria per seconds are released. It should be recognized that in a cooling tower, not only does evaporation of water occur, but along with it, there is a formation of water droplets are also occur. Now this cooling tower spray the water droplets, and if it is contaminated, then there is a then it will become a possible source for a certain pathogen and consequently airborne contamination. Microorganisms and small particles are commonly found in a immediate vicinity of water surfaces. Now the next is your outdoor environment. The control of airborne organism in the immediate surrounding of dairy premises is more difficult than in a closed indoor environment. Okay, if we have to control the airborne microorganism in an immediate surrounding, that is your open surrounding of dairy premises, it is very difficult as compared to the closed indoor environment where more control measure can be taken. Okay, if we have a closed environment, then we can control the airborne microorganisms very easily. Okay, now according to Al-Dagal and Fung 1990, one aspect that could be helpful in reducing the microbial load in outdoor is the control of the organic material. When we control the organic material, then we ultimately we control the microbial load in the outdoor environment. Now different natural agents will be used for controlling this organic material. These are what? These are the UV light, humidity, temperature, wind direction, and also speed have significant influence on the total number of airborne microorganisms in the outdoor atmosphere. Now the next is your processing room. The most positive approach to controlling the airborne contaminant indoor is to remove all the contamination source from the area where the product might be exposed to a air. So when we have to control the airborne contamination inside, in that case we have to remove all the contamination source from that area where the product are come in contact with the air. Good ventilation is very important. Okay, to remove the moisture which is released during the processing of the dairy product. Okay, when there is a good ventilation present in that area, then it will remove the moisture which are released from the processing of the dairy product. It will also prevent the condensation, subsequent mold growth on the surface. More attention is given nowadays to a air cleaning in food plant, among other things, by establishing airflow barrier which are against to a cross contamination from the environment. In modern dairy plant, the air entering processing room is chilled and filtered to remove a particularly air, all bacteria, yeast and mold. It is very essential that filter and the sterilized air should be supplied to that area where the sterile operation are carried out. Okay, when we have to carry any sterilized operation, then at that area the air sterilization and the filtration of the air are very important. So that's why we have to apply the filters and the sterilized air at particular that area. Okay, now the next is your rigid frame filter are closely packed glass fibers are available which is achieved to a contamination free air for the culture transfer, manufacturing and packaging of the sterile milk and milk product. So for this purpose what we have to do, we use a high efficiency particulate, that is your HEPA filter, so that it can remove the around 99.99% of the airborne particles 0.3 pm and larger, while new ultra HEPA, that is your alpha filter, it will remove around 99.999% of particles as small as 0.2 pm. Passage of air through a combined HEPA or alpha filter is usually considered suitable for the use where contamination free work is to be carried out. Okay, so at that area where contaminated free work should be carried out, at that area we use a HEPA or a alpha filter, so that HEPA filter will remove the 99.99% of airborne particle, and it can also remove the 0.3 pm size of the microorganism, and alpha filter can remove the 99.999% of the particles along with a as small as 0.12 particle size of this microorganism. Standard high efficiency air filter system allow more air into the room than a normal, thereby establishing a positive air pressure upon the opening a door filter or a air flow out, thus blocking the entry of the untreated air and minimizing the microbial contamination. For optimal ventilation, sufficient air changes have to be made which prevent the buildup of the condensation on the surface, and this will achieve by a not less than 10 changes per hour or often to a 20 changes per hour in a room where moisture is generated. Okay, now the next is your room in which direct exposure to outside air is inevitable can have airflow barrier installed mounted over open doorways to secure a significant downward velocity of the airflow, preventing or contamination from the outside. And this outside source should be filtered with a free of the condensate. Then compressed air should be commonly used in various processing operation, and it can be contribute to the contamination of the product by dust and microorganism in the case of lubricated compression system by a oil fume. Whenever air under a pressure come into a direct contact with the product or is directed at a milk contact surface, it should be of the highest quality. Now, we can also use a sterilized compressed air, and this sterilized compressed air can be obtained by drying the air after the compression in adsorption filter. Example, pure cotton, polyester or polypropylene are used. And by installing a series of filter with size of 0.2 ppm pore size downstream, immediately it will preceding the equipment where the air is needed. Okay, now the walls and the ceiling must also be of the highest standard with no opportunity for the accumulation of the dust and other deposit. To achieve the required standards, suitable sealant and sealant as well as coating with the antimicrobial properties are available with effectivity against a wide range of bacteria, mold and yeast. The use of the clean room clothing, head covering mask and gloves largely eliminated the release of the microorganism into a processing environment. Okay, if clean room clothing are used, okay, head should be covered, mask should be there and gloves should be there, then ultimately what they do, they eliminate the release of the microorganism into a processing area. A good hygiene training program for a factory personnel will contribute to reduce the contamination by the worker. Okay, so this good hygiene training program should also given to the employee who are engaged in the work of work for the work which are related to the production or the anything else which are like workers who are working inside the industry. So all the workers should take in the good hygiene training program, and it will also contributing in reducing the contamination by the workers. Standard for a airborne corn count in various processing area have been proposed by our various authors. Now, proposed standard in this regard are present in a table. 14.1 will be discussed in a next in a further slide. Although these standards are relatively strict, experienced and shown that they are achievable. Methods for the air sampling. So the main reason for the air sampling in the dairy plant is to evaluate its quality and also obtain the information about the hygienic condition in certain clinical area where the microorganism may contaminate the product either directly or either indirectly. And this sample is taken in by a following way, like at the opening of the processing equipment that may be subjected to a potential contamination by a air current at a selected point in a room, for example where a product are filled and packed, and the area where the employee are concentrated. So we take a sample of we take a air sample from a three location of this industry, dairy industry. First, the opening of the processing equipment, which are subjected to a potential contamination, and the second one is the point of the room are selected where the product are filled or up, and the third area is where the employee are concentrated. Now this is the table for the standard of the air count in various processing industry. Like if we have a processing area for a cultured milk and cream and cottage cheese. Okay, so the plate count, if we talk about the plate counts, satisfactory value, then it will not more than 150 CFU. If it is not more than 150 CFU, CFU means that colony forming unit, then it is satisfactory. And it is unsatisfactory if it is more than 1500. Okay, yeast count less than 50 and unsatisfactory if it is more than 100. Now milk and milk cream, again it is less than 150, then it is satisfactory, and more than 1500, then it is unsatisfactory. Here it is for a plain count, and here it is for the yeast and mold. For a butter, if it is less than 100, then it will be satisfactory, and if it is less than 1000, then it is unsatisfactory. Powder milk, if it is less than 200, then it is satisfactory, and if it is more than 2000, then it is unsatisfactory. Ripe cheese, if it is less than 200, then it is satisfactory, and less than 2000, then it will be unsatisfactory. Now, ripe cheese in case of yeast and mold count, if the colony forming unit less than 100, then will be satisfactory, and if it is more than 1000, then it is unsatisfactory. So these are the standard for the air count in various processing industry for a plate count and along with a yeast and mold. Although various air samplers have been designed for sampling airborne organisms, none of these recover viable particles without some inactivation or losses during or after the sampling. The effectiveness of the air quality monitoring totally depend upon the type of the sampler which are used, and as well as on the nature of the air in a specific environment to be monitored. Now there are two main principles by which these airborne microbes can be sampled. First one is your collection on the solid and semi-solid media or a filter, or collection into a liquid solution or a medium. The objective in each case is to determine the number of the organism on the plates, in the filter, or in the liquid media. Sampling time for all the collection method is usually sterilized at 50, 30 and 60 minute IDF 1987. The basic method is include sedimentation, impaction on solid surfaces, and impingement in liquid, as well as centrifugation and filtration. Comparative studies for air sampling devices has been indicate that there is often no obvious choice of the correct samples to be used, and the result in this regard are discussed by Kang and Frank in 1989 A2C and 1990. So this is all about the like this is all about the today's lecture. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on your on YouTube, and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide, that is 70808334 450 and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel. Thank you so much. Again, if you have any query, just let us know in a comment box.